Okay, next, housed in the scrotum, we have the testicles. Testicles or testes, either name is acceptable anatomically. The testicle, about the size of a walnut, no matter what us males like to say about the size of them. Not very large structures. Walnut is a pretty dang close scenario of being the same size. Now the testicles are divided up into lobes, and within these lobes are these, and of course I can't draw it very well, but in these lobes you have these coiled up tube systems, very complex tubule systems in here. Now these tubule systems are known as your seminiferous tubules. And it is these tubules in which gametogenesis occurs, or spermatogenesis. Spermatogenesis is just the fancy name or fancy word for the creation, because genesis being creation, of our sex cells or our sperm. And this complex coiled tube system, even though they're only about the size of the walnut, we have about 18 foot of coiled up tubule system to produce our sperm in this way. Now, once the sperm migrate from the outside of the tubule toward the center of the lumen of the vessel, they begin to migrate down the tubules to an area on the testicle that we have another structure that we call the epididymis. Now, the epididymis is this structure here. Let me write over here. Epididymis. I may have misspelled that. Maybe the Y's there and the I's there. I don't know. Maybe I'm screwing up the spelling of that one. But the epididymis is basically the place that we store the sperm. They haven't quite completed maturity. They actually don't complete maturity until actually after they've been ejaculated from the male's body. But here's where the sperm that are ready to be delivered in an ejaculation are stored and housed. Now, if the male has an ejaculation, the smooth muscle contractions of the epididymis pushes the sperm out of the epididymis up the ductus deferens, which this tube here is the ductus deferens. If you're old school, you may remember that the old name was the vas deferens, but the ductus deferens is the new name for the tube that then connects the epididymis onto the urethra, where the male, which will come through this tube right here and dump into the urethra. Now, once that dumps into the urethra will then propel the sperm on out the end of the penis for the male orgasm or the ejaculation. Now, the ductus deferens is completely lined with smooth muscle, same as the urethra there. So it is these peristaltic contractions that can move the sperm on down. But we still haven't mixed the sperm with the liquid parts of the ejaculate yet. The liquid parts of the ejaculate or the semen come from contributions from other structures. And the two major structures are the seminal vesicles, which a lot of times people get confused in their introductory classes to A&P, with the seminiferous tubules, which are in the lobes of the testicle, where they produce sperm, and the seminal vesicles that are located here on the back of the bladder. So if we pull the bladder out of position and look on the back, you will see one on this side and you'll see one on the other half of the bladder. So running in on the back side of the bladder, we're talking about something a little smaller probably than your little finger, angling inward here with the tubule system, runs into the prostate and dumps into the prostate here in the urethra, running through the center of the prostate in the male. And so the seminal vesicles are going to contribute the liquid portion, or some of the liquid portion, liquid or semen, to the ejaculate. And they are going to be assisted in this with the prostate. And I just showed you how the seminal vesicle runs, the ejaculatory tube that runs through the prostate is through the seminal vesicle connecting to the urethra. And so the male prostate is also going to do about the same thing. Now, the contributions are going to be slightly different chemically. Each one has its own functional parts because there is a whole complex. It's not just a liquid material like water. We're talking about complex proteins like what they call fibrinogen-like proteins, which helps make the ejaculate sticky. Because when we release the ejaculate inside the female's body, when we're having penetration with intercourse, we want the ejaculate to stay where it lands. We want it to be fairly slow to succumb to gravity to drain out of the female's body. So these sticky proteins are very similar to the same proteins that form the coagulation aspect with blood clotting. We find these proteins in the semen. We have other materials like citrate, fructose. Again, one of our important monosaccharide sugars we typically get from fruit sources. But it is this material that gives the energy to the sperm. The sperm have mitochondria, very large mitochondria in the midpiece or the neck of the sperm itself. When you look at a sperm, it's in this neck midpiece that we will find these large mitochondria. And we find that the fructose in the liquid material is the source for making ATP so that we can do the whip-like action with the flagellum so that the sperm can propel themselves. Because they have to do pretty much a marathon swim if they're going to reach the ovum and possibly fertilize the structure. So there are several things. In fact, I guess now that I got sperm drawn up there, I would like to point out that this structure is the acrosome. Very important cap-like structure on the head of the sperm. Now, in here is where we contain the 23 chromosomes, because remember, dad is gonna contribute 23 chromosomes, mom's gonna contribute 23 chromosomes, and when this genetic material fuses in the nucleus of the ovum, we will then basically start a new human life. And so the acrosome is a very important structure because it contains enzymes which can help digest a couple layers off of the ovum. The ovum is protected. So if an ovum is huge compared to a sperm, but they have this layer system here known as the zona. The corona radiata is out the first one. Corona radiata and the zona pellucida. And it's these layers that have to be eroded away. So as the sperm comes in and releases its enzymes from the acrosome, it actually will digest a portion of that layer away. Let me see if you guys can even see that. Oh yeah, okay, good. I was just making sure. I didn't know if I could see this part of the board. So therefore, if we only have one sperm reach the ovum, we'd never get that girl pregnant. Usually it takes somewhere between about 50 and 100 sperm to produce enough enzymes to erode enough of this layer away that one sperm can fertilize it. Now, as soon as one sperm touches or crosses the membrane of the ovum here, so once it erodes that down and one penetrates across, there's almost an instantaneous cortical reaction in the ovum to prevent any other sperm from penetrating. Because we can't have 23 more chromosomes getting in there and messing things up. So this instantaneous cortical reaction helps prevent what we call polyspermy, or more than one sperm from fertilizing the egg. Now, once we have the sperm penetrate that, its genetic material will then migrate through the cell to the nucleus, and combine to form the proper 46 chromosomes in a human cell. And so that is why it really takes more than one sperm. Yes, only one to fertilize it, but it really takes at least 50 to 100 reaching the ovum to be able to do that. And on average, males produce about 300 million sperm per ejaculation. This seems quite a lot, but it really takes a lot to get a few hundred to even make it up through the uterus, through the cervical canal, through the uterus, up the fallopian tube to reach the ovum. For successful fertilization, we typically have the sperm reach the ovum still in the fallopian tube, so that it has gotten to the blastocyst stage by the time it reaches the uterus, so the blastocyst can implant in the blood-rich nutrient layer that we call the endometrium. Now, even though I said 300 million sounds like a heck of a lot, believe it or not, and I'm really curious to see if there's been any studies since then, but in 1940, they did a large sample size of males and found that the average male was almost twice the sperm count, around 600 million sperm per ejaculation. 50 years later, 1990, that's where this statistic comes from. 1990, the average male only produced about 300 million sperm. Now, what is causing this lower virulence rate or lower fertility rate in humans? Lots of factors probably, but only speculation. Nobody really knows the answer here. But if we keep on this trend, it's going to be very difficult for males to get females pregnant, and if our reproduction stops, yeah, that's the end of our species. So maybe we ought to spend a little money and time to figure out what's going on there. All right, we'll start with that next.